Diego, can you unmute?
Yeah, I wanted to say hello. I'm Diego Restrepo from Cell Developmental Biology, CU Anschutz. And in the last 10 years, there's been a revolution in understanding the brain by recording neural activity, recording calcium changes. But this is a major limitation because what you really need to record from neurons is the voltage. So, and what we want to do here is to make a faster imaging voltage sensor for freely moving animals. And this is a group that has worked in the past. We have a CU Anschutz bioengineer, Dr. Emily Gibson, Avi Persson, who is a professor in physiology. In CU Boulder, we have Dr. Victor Bright, who is in mechanical engineering, and Dr. Julie Gopinath in electrical engineering. And what we do is basically make little miniature fiber coupled microscopes. So this was published about 2 years ago under the NIH Brain Initiative. And what we'd like to do is to modify that microscope to do really fast measurements of voltage at 1 kilohertz. And the technical approach is basically to do high-speed imaging using a sensor that's called ASAP3. And what we really need is to incorporate a computational imaging scientist from Boulder. The milestones would be to record at about 3 kilohertz, signal to noise ratio of bigger than 10 hertz, 10-fold. And it's to understand the brain function. So that's basically it. Thank you.
Thank you. Next, we have David Root.
Thanks, Kristen, and thanks to all of the organizers. My name is David Root. I'm a fairly new assistant professor in the Department of Psychology and Neuroscience in Boulder. And we're interested in identifying different cell types in the brain's reward center that control different aspects of motivated behavior. So that is your everyday experiences of reward and aversion, but also in extreme states such as addiction, stress, and depression. We've focused on the reward center ventral tegmental area, and we've discovered that there are more cell types than we previously thought there were. And we've developed methods in the mouse to target these different neurons. We've found that they have different projections throughout the brain. They seem to be sensitive to different cues and experiences involved in reward and aversion. But what we're looking for now is a collaboration trying to understand more about these different cell types. Specifically, we're looking for electrophysiologists that could perform whole cell recordings of these cells, or even those that can perform RNA-seq experiments, trying to figure out what kind of receptors each of these cells have, what transcription factors they express, et cetera. We've been lucky to receive a number of awards looking at these different cell types in the VTA for different aspects of motivated behavior. And all of the research that I'm proposing in the collaboration can lead to new, likely NIH funded applications. So please get in touch if you're interested.
Thank you, David. Daniel Denman is our next presenter.
Thanks, Kristen. Hi, everyone. My name is Dan Denman. I'm in the Department of Physiology and Biophysics at Anschutz. I'm a new assistant professor there. I've been here on campus for less than a year. I run a basic neuroscience lab here. Our general question is how neural populations underlie all forms of behavior and especially perception. So the way this is typically studied is one neuron or one area at a time, but it's well known that the sort of number of neurons that are involved in generating even a very simple behavior in a mammalian nervous system is on the order of millions and more likely tens of millions. And so we take an approach that attempts to embrace this complexity and record sort of from as many neurons as we can while animals are performing a visual behavioral task. And so we use a couple of devices that I developed in my previous work. One is an immersive virtual reality system that allows us to record invasively in mouse brains while an animal is performing a task. That's shown there on the left of the schematic, sort of animals in a virtual reality dome. The other is an electrophysiology device, which is essentially a small array of electrodes in which we can put many small arrays of electrodes in the brain. And so see a schematic of about 12 of those electrodes stuck in a rodent brain. Each one of them can record several hundred neurons. So we're talking order several thousand neurons at once. And our goal here in looking for collaborators is one, always interested in developing new devices. As I said, I helped develop those electrophysiology devices in my previous work. And so looking at new ways to record from large scale ensembles of neurons throughout the brain is a big part of our research, but especially dealing with the sort of scale of analytics and quantitative modeling of this type of data is our goal in seeking out collaborators. So looking at computational models, applied mathematicians and physicists who are interested in working with this data. Currently funded by the